All right. Well, thank you so much, Dr. Antonescu. That was a great presentation, and appreciate you going through all those questions with us. And I don't know if you guys noticed this when Dr. Antonescu was on the screen or not, but I love your pin. Thank you for wearing proudly the EHE Just Live pin. We appreciate it. So we started off the day with Dr. Tapp and Dr. Antonescu, again, our lead clinician and researchers on the CZI grant, and just tremendous appreciation for their dedication. So thank you. Next up, I am excited to introduce Dr. Valerie Kouskoff with the University of Manchester, UK. Dr. Kouskoff will be presenting on an in vitro model to study the consequences of TAS-CAMTA1 expression in primary endothelial cells. I practiced that about 5 times last night, so I'm glad I did it without messing up. We appreciate her dedication to EHE research, and please welcome Dr. Kouskoff.
Okay, well, hi everybody, and first I would like to thank the organizer for giving me the opportunity to present the data we are doing, the work we're doing in my lab. So my lab is really basic biology, and we started this project about 2 years ago, and so we're trying to develop an in vitro model to study the consequence of TAS-CAMTA1 fusion protein in endothelial cell. So the TAS-CAMTA1 is the same as the WWTR1 CAMTA fusion protein, but for simplicity, I use the TAS-CAMTA1. So to do this in vitro model system, we use embryonic stem cells. And embryonic stem cells are cells that are taken from the inner cell mass of a blastocyst embryo, the 5 embryo, and they can be expanded in vitro, and we can keep them for a long time in vitro. And those embryonic stem cells are really a powerful system to study a lot of developmental process. So embryonic stem cells, they can be maintained and expanded in vitro, so we can generate lots of those cells. They can be changed by genetic manipulation. But what is really remarkable about ESL is that you can differentiate them in culture to generate all type of cell from the organism. In my lab, we are particularly interested in studying cells that are mesoderm derivative, which include the endothelium, blood cell, heart, and muscle. So how we do that? So we take embryonic stem cell and we induce them to differentiate. And when they differentiate, they will form those 3D structure that we call embryoid body, and in which all type of cells can develop. And from those embryoid body, we can isolate progenitor cell. Here they are marked by expressing a FLIC1 molecule. And those cells can be further cultured to generate endothelial-like vessel structure. So through several steps of culture, we can generate angioblast and endothelial culture. So to study, we really became interested in studying how TAS-CAMTA1 can modify or affect those endothelial cells. So to study and to express TAS-CAMTA1, we generate an ESL model system in which we can induce the expression of TAS-CAMTA1 protein by doxycycline. So without doxycycline, the cell do not express TAS-CAMTA1, but when we add doxycycline to the system, to the cell culture, they start to express TAS-CAMTA1. And to make our life easier to study the cell that express this fusion protein, we link TAS-CAMTA1 to a fluorescence protein, GFP, which stands for green fluorescence protein, and also the TAS-CAMTA1 contain a flag sequence that we can use antibody to recognize this flag sequence. So we got this construct from Brian Rubin's lab, and we engineered those embryonic stem cells in which we can now study the consequence of TAS-CAMTA1 expression. So using this model system, we can monitor TAS-CAMTA1 expression with various tools. So we can perform flow cytometry, and here what you see are cells that either without doxycycline or with doxycycline. And when we add doxycycline into the media, what we see is that a lot of the cells start to express GFP. And those cells express GFP, but also express TASCAM-T1. We can follow TASCAM-T1 by immunofluorescence. So here you see cells that are stained for their cytoskeleton, but also with flag, which tag the TASCAM-T1, and we can see here it's that the TASCAM-T1 is in the nucleus of the cell. We can also perform Western blotting, either using anti-flag antibody or anti-TAS antibody, and with this we can study protein presence and protein interaction with the TASCAM-T1 fusion protein. So we have a variety of tools to start studying what TASCAM-T1 is doing. So the first step in this project was to establish an experimental protocol in which we could grow endothelial cell and induce the expression of TASCAM-T1. So we started with those inducible TASCAM-T1 embryonic stem cell. We induce a 5-day differentiation in which we generate embryoid body. At day 5 of this culture, we harvest all the embryoid body and we sort for an endothelial progenitor. And those cells are isolated based on the fact that they express FLIC1 and TI2, which are two molecular markers of endothelial progenitor. Then those cells, which are purified from the embryoid body, are put in endothelial culture. And at various days, we can induce TASCAM-T1 expression with doxycycline and we can perform analysis. So this is the culture system, how it works. So this is a day 5 embryoid body. So by flow cytometry, we can detect those cells that co-express FLIC1 and TI2. So by flow cytometry, again, we can sort those cells and put them in culture. And what you see here is the expression of various markers that are expressed by endothelial cell, TI2, FLIC, but also CD31 and V-cadherin. And at day 3, you have a mixed population of cells that express various levels of those different markers. But by day 7 of this culture, what you see is that most of the cells express high level of FLIC-positive, FLIC1 marker and TI2, and moderate level of V-cadherin and CD31. So we have a fairly homogeneous cell population in those cultures starting from day 7. And in those experiments, what we did is to try different substrate on which those cells are cultured, gelatin, fibronectin, or matrigel. And further experiment revealed that matrigel was the best substrate to grow the endothelial cell. And here you can see a photograph of those cells at day 5 when they are cultured on matrigel. And what you can see is that those cells start to organize into a tube-like structure. So that's the beginning of the organization of those cells into a vessel-like structure. So once we have this experimental protocol set up, so we started to investigate the impact of TASCAM-T1 expression on various parameters. So the first one we looked at was the endothelial cell identity and whether expressing TASCAM-T1 affect this endothelial cell identity. So here what you're looking at is, again, flow cytometry. So doxycycline was added at day 7 of those culture, and at day 8, 9, and 10, what we looked at is the expression of GFP. And what is clearly visible here is that we can detect two types of expression level, cells that express low level of GFP, and cells that express high level of GFP, and cells that do not express GFP. So the first observation was that we don't really see any increase in the frequency of cells that express GFP low or GFP high. It's fairly constant, and if anything, this frequency of cell decrease over time. So the next thing we looked at what is the expression of endothelial marker on those cells? And here you're looking at that panel, and this is looking at the cell that express high level of GFP, therefore high level of TASCAM-T1, low level of GFP, or that do not express the fusion protein. So without expression of the fusion protein, as I showed earlier, the cell are fairly homogeneous in the expression of the endothelial marker tied to FLIC1. But what is striking is that as the GFP level increase, what we see a decrease in the expression of those endothelial marker, and GFPI expressing cell are very low expression for those endogenous markers. So what it seems to be is that when TASCAMTA1 start to be expressed in those cells, they lose the expression of endothelial marker. So currently we are doing further experiment to try to determine the extent of the change in the endothelial identity of those cells, and we're doing that through global transcriptomic of those cells. So next, what we looked at is the impact of TASCAMTA1 on cell death and cell proliferation. Because what we observed is that the cells that express GFP and TASCAMTA1 don't seem to change much in their frequency. So the first thing we looked at is cell death. And we can measure the amount of cell death by staining the cell with annexin 5 and propidium iodide. And what you are looking at here is flow cytometry for those two markers. And the cell in this quadrant are live cells, whether cells that are in this quadrant are necrotic and cells here are apoptotic. And what we see is whether the cell express GFP and TASCAMTA1 or not, or they have not been induced by DOX. We don't see any differences. So it doesn't seem that in this experimental system, TASCAM doesn't induce any increase in cell death. So the next thing we looked at is cell proliferation. So the way the proliferation cycle of cell is divided into 4 phases. So you have a quiescent or resting phase called G1-0, an S phase in which the cells start to synthesize DNA in preparation for cell division. So the cell after the S phase move into the G2 phase, and here they are preparing for mitosis, and M is the mitosis phase of the cell cycle when the cell divide. So to measure the amount of cells that go into those various phases, we can use the incorporation of EDU, which is a small molecule that will integrate into the DNA. So the experimental protocol was to take the endothelial culture at day 10, add doxycycline and induce the expression of TASCAMTA1 for 24 hours, then to add the small molecule EDU for 4 hours, and then to analyze the cells. So when we did that, what we observed is that, so this is looking at the various phase of the cell cycle. Those cells here in the orange gate are in G1-0, so they're resting. Those cells in the green gate, they are in the S phase, and those cells in the pink gate are in the G2M. So what we see, and here we look at the flag expressing low and flag expressing high cells, what we see is that many more cells, the frequency of cells that are in the S phase is really increased in the flag expressing cell. And this is a summary of a lot of experiments showing that we see a statistical decrease in the number of cells, in the frequency of cells that are in the G1, G0 phase, and a strong increase in the frequency of cells that are in the S phase. Very surprisingly, we don't really see any increase in the frequency of cells that are in the G2M stage of the cell cycle. So what we can conclude from those experiments is that the expression of TASCAMTA1 pushed the cell into the S phase of the cell cycle. But what we don't know right now is why they are not progressing further into the G2M. So we are performing further experiments to try to understand why those cells are blocked into the S phase. But it certainly fits with the idea that we don't see any increase in the frequency of cells that express TASCAMTA1 in our endothelial culture. So another aspect of the TASCAMTA1 regulation that we started to look at is how TASCAMTA1 might be regulated by external cell signaling. So we know from a lot of work by many groups that TAS, the wild-type TAS, is regulated by multiple pathways. So there is the HIPPO pathway, which includes cell-cell contact, attachment to the extracellular matrix, or glucose deprivation. But TAS is also controlled by mechanical force, and this is outside of the HIPPO pathway. So we were interested to start to understand whether TASCAMTA1 is also regulated by mechanical force. So I'm just going to explain a bit what what mechanical force are and what we are doing to try to address this. So mechanical force can be shear stress, compression, stretching, stiffness, adhesiveness. And this is mediated by the cell-cell interaction or by the extracellular matrix interaction. The microenvironment of the cell is very important in those mechanical forces. So mechanical stimulation impact directly the cytoskeleton of the cell, and this will in turn impact many aspects of the biology of the cell, such as proliferation, differentiation, cell fate decision. So the type of change in the cytoskeleton we looked at are change in the conformation of the actin, which form the cytoskeleton, change in the binding of protein to those actin fiber, and also change in the way the actin fiber polymerase. So as a first approach, what we did is to use inhibitor of this change in the cytoskeleton. So here is a table with the list of the 3 inhibitors we've used so far. So blebistatin that interfere with the myosin, and the myosin are the small molecule that form the F-actin filament of the cytoskeleton. And we also use latrunculin B and ROCK inhibitor, and those 2 inhibitors affect the polymerization of the actin filament. And all those inhibitors have been shown to affect the function of wild-type TAZ in normal cells. So the experimental protocol here was to take those D10 endothelial culture to add doxycycline to induce the expression of the fusion protein TAZ-CAM-TA1, and then 24 hours later to add the inhibitors for 28 hours, and then for 4 hours, and then to analyze the outcome of this culture. So here by flow cytometry, we are looking at the expression of the flag TAZ-CAM-TA1 in those cells after 4 hours of inhibitor action. So this is the control with no inhibitor added. What you see, a large fraction of cell express high level of flag and a smaller fraction express low level of flag. When the cells are treated with either of those 3 inhibitors, what we see is the frequency of that express high level of the flag decrease, whether the cell that express lower level of the flag increase. So certainly the inhibitors are doing something to the stability or the expression of the TAZ-CAM-TA1 protein fusion. But we looked further at how this treatment with those inhibitors may affect the cellular localization of TAZ-CAM-TA1. So here in the upper row, what you see is the control cell culture, and phalloidin here is staining the actin cytoskeleton, and here what you see is all staining for the cytoskeleton in endothelial cell. The flag antibody stain for the TAZ-CAM-TA1, and you can see here is that most of the TAZ-CAM-TA1 are located in the nucleus, which you can see with DAPI staining, and this is a merge of these 3 staining. So in the control culture, most of the TAZ-CAM-TA1 protein are located within the nuclear, where they are effector of transcriptional program. But when we treat those cells with any of those 3 inhibitors that modify the architecture of the cytoskeleton, and you can see here the cytoskeleton, the staining for the actin is very much affected by the treatment. What you see, and it's very much visible with a blebistatin staining, is that now the flag staining is still found a bit in the nucleus, but mainly it is found in the cytoplasm of the cell. Here you can see where the nucleus here for this cell, and you can see here in the merge staining that a large fraction of the TAZ-CAM-TA1 is now located in the cytoplasm of the cell, and we can see the same for the 2 other treatments. So those data indicate that the cellular localization of TAZ-CAM-TA1 is most certainly regulated by mechanical forces, and we're starting to perform other type of experiment to trying to really address how this is performed at the molecular level. So this is a final slide with a take-home message. So in my lab, we have established an embryonic stem cell doxycycline inducible model system to study the impact of TASCAM expression of endothelial cell. We established a robust protocol to generate endothelial cell culture. And the data so far seems to show that TASCAM-TA1 expression affect the cell surface expression of endothelial marker. And as I said earlier, now we're in the process of performing global transcriptomic analysis to see what are the change over all the program of the endothelial cell that are affecting the expression of TASCAM-TA1. What we have observed is TASCAM-TA1 promote the entry into the S phase of the cell cycle in endothelial cell. And what we want to understand now, why are those cells stuck in the S phase? And so what happened to the regulator of the cell cycle when TASCAM-TA1 is expressed? And finally, the third area of research we are pursuing is trying to understand how the expression of TASCAM-TA1 is controlled and regulated by mechanical forces. And this is particularly important in the context of the tumor cell, as we know that the microenvironment is very important to guide the progress of the tumor cells. So we are now looking at how the microenvironment affect TASCAM-TA1 function in the endothelial cell culture. So to finish, I just want to thank Emily Neal in the lab. So all the data I presented were done by Emily Neal, a PhD student in my lab, and she is funded by a fellowship from the EHE Rare Cancer Charity UK. And I thank you for your attention, and I'm happy to take any questions.
All right, we've got a couple of questions that have come in. The first one relates to the mechanical forces that you talked about most recently here. So the mechanical forces experiences by EHE at different anatomical locations, so for example, pleural versus liver, are quite likely different. The question is, can you test the effect of these different forces to help tease apart the different behavior of EHE across different sites? And what are your thoughts on that?
Yeah, I think it's a very important question, and I think we have to first try to define what are those different mechanical forces in each of those organ, and certainly taking a cell from this organ or putting the endothelial cell in mouse model system in either liver or lung would help to understand the behavior and how the TAS expressing cell react to the microenvironment. But definitively this in vitro system offered the opportunity to dissect those mechanical force, and also how this might result in a transition from an indolent disease into an aggressive disease.
Excellent, thank you. I guess this other one kind of relates to mechanical forces as well, but I suppose it's also kind of directed towards possibilities and improvement of the in vitro model. The question is, have you done anything in terms of tuning the stiffness of the matrigel to regulate expression of TASCAM, or played around with the sort of the surface conditions?
Yeah, in fact, that's the next series of experiment that Emily will start. Is we have plates, tissue culture plate of different stiffness on which we're gonna. Emily is planning to coat them with matrigel and then to put the cell and then induce the expression of TASCAM-TA1 and see how different stiffness are likely to affect the biology of the cell or the behavior of the TASCAM-TA1 protein.
Thank you very much. Got a question here about whether your model has revealed anything about or any insights about the cell of origin of these EHE cells that we would see in vivo, since you've taken basically an embryonic stem cell and advanced it to a EHE-like cell. Has any of that process revealed any clues about what the precursor cell would be for EHE?
I think it's a difficult question. What we see is that, so we've done previous experiment that I didn't include, but if we try to express TASCAM-T1 earlier, like in early mesoderm or in really angioblast, we see a very different sort of behavior, but it doesn't seem that we can maintain the cells. So I'm not sure it really addressed the cell of origin. It's probably not the best model system unless we can really find a way to transition endothelial cell expressing TASCAM-T1 into sort of a tumor model. I'm not sure we can really address this question.
Yep, that's a tricky one, and one I think you'd have to do the experiments to see. Got one more question here that's come in. If you allow the TASCAM induction with the doxycycline to grow past day 10 of culturing, do you see greater change in the percentages of flag high, flag low cells in the population?
So the problem we have, well, not the problem, but with this system, what happened is that at some point the cell culture reach compliancy, and the cell touch each other and they stop growing. And that's something also that will be interesting to study, is what happened when there is so much cell-cell interaction. But for the moment, we really have not investigated that too much. But I think, if I remember correctly from the data from Emily, is that the frequency of cells that are GFP positive remain the same throughout the culture.
Excellent. And then is this a cell, an in vitro model, when you have these cells that could possibly be transplanted into an animal model to have basically a xenograft model for analyzing effects the animal system might have on these cells?
Yes, this is something we would like to do once we really understand the system. It's certainly something that would be very interesting to see, to perform, to put those cells in like matricial structure and to implant them in different parts of different organ of the mice. That's something that we have in mind, and it might be a really good system to test potential drugs or this kind of thing. So this is something we want to develop in the future.
And related to drug testing, and if this model ends up becoming quite important for that, and cost issues and possibly regulatory issues, do you think a similar model could be developed from induced pluripotent cells rather than embryonic cells?
Yeah, that would be really great. I think that's something also, it's not part of this project we are doing, but that's something I have in the back of my mind, is to try to derive induced pluripotent stem cell from patient samples. But right now we really don't have the manpower or the money to embark into this type of project. But that definitively would be a very interesting model system to use.
Well, we'll have to keep that in mind for looking for future funding and resource opportunities for you. That's all the questions that have come in. Thank you so much for your time today.
Well, thank you very much.